So hi, I'm Karen and I'm currently an intern at the Gilmore Lab, which is a molecular biology laboratory that's at Boston University. So the research that goes on in this lab mainly focuses on a protein known as NF-kappa B. This protein is in charge of expressing specific proteins that are very important to our immune and inflammatory responses. So the way that this protein is activated is through a receptor that is found on the cell's membrane, and so a molecule on the outside of the cell will bind to the receptor, and then a cascade of proteins will go down all the way to where NF-kappa B is. And as I said before, it'll start the expression of proteins that are very important to our immune and inflammatory responses. So the first proteins that interact with this receptor are MAL and MyD88, and they're extremely important, and without these proteins the whole cascade process would not be able to work. Anyways, this pathway is found in organisms from humans all the way to flies, but more recently it's been identified in older organisms such as jellyfish, sea anemone, and the endangered coral Orbicella favulata. And as I said before, this pathway has been identified in Orbicella favulata, and so one of the goals of the lab is to be able to take the toll-like receptor on the coral cell and then put it in an environment where it could potentially interact with MAL and MyD88, which are the essential proteins in order for the cascade to occur, and to hopefully see the pattern that we've been seeing in human cells. And so this research is very important because we firstly do this for the sake of science. We look at the molecular process that happens in older organisms in order to understand our immune system. And also, in addition to that, we do this for the endangered marine life. As I said, the coral Orbicella favulata is one of the endangered corals due to global warming, and just like any other coral, it is very sensitive to its environment, whether that be a change in temperature, light, or nutrients, and this can leave it very susceptible to diseases that could kill it. And through this research, we want to be able to potentially aid these organisms as they undergo stress.